Welcome back to MLT 240. In this lecture, we'll be covering mycology. This lecture will be divided up into 3 parts. So what is mycology? Mycology is the study of fungi. This is section 1, the introduction to mycology. The next 9 slides, slides 3 through 11, are all covering terminology you need to know and be aware of for the mycology unit. For time's sake on this video, I'm not going to take time to read through each one of these terms, but please go through on your own in the PowerPoint, read through these terms and become familiar with them. For the video purposes, I'm going to skip down to slide 12, which is the introduction. The importance of mycology and studying mycology is increasing as the number of immunocompromised patients increase in our population. Fungi is classified as either yeast or mold, or pathogens, or saprobes. Saprobes are a group of fungi that are decomposers that feed on dead or decaying matter. Within the kingdom fungi, fungi are eukaryotic, like plants, with a nucleus, a nuclear membrane, and a mitochondria. Unlike plants, fungi lack chlorophyll and must absorb nutrients from the environment. So really, on a molecular level, fungi are more like animals than plants. So just so you get an idea of the classification for the kingdom fungi, let's look at this diagram, or this image. Everything that we've talked about in Unit 1 for our amoebas falls in the kingdom Protus. Okay? Everything in Unit 2 for the nematodes falls under the kingdom of animals. But everything in our mycology unit is going to be falling under the kingdom of fungi. The shape and structure of fungi are maintained by a rigid outer cell wall. This unique cell wall is composed of chitin, which is a sugar molecule that is made by some plants and animals, glucan, which is a polysaccharide, and polysaccharide protein complexes. And you can look over to the right hand side and you can see the basic structure, and you can see the chitin, the proteins, the glucan that makes up the fungal cell walls. Most fungi are obligate aerobes that grow best at a neutral pH, but some can tolerate a wide pH range. Fungi need moisture for growth, but spores and conidia can survive in dry conditions for extended periods of time. And spores are cells that are involved in fungi reproduction, and conidia is spores that are produced at the tip of hyphae, or branching filaments of fungus. If you look here to the right, you can see some spores have been released from this mushroom or fungi. So they're just small little cells that help for reproduction of the fungi. And we'll see an example of what conidia looks like on the next slide. Fungi are non-motile. Before we get into the differences of yeast and mold, I just want to point out these little small balls on the end of this hyphae are examples of conidia. Yeast is usually a single vegetative cell that usually forms smooth, creamy, bacteria-like colonies without aerial hyphae. So aerial hyphae means things that grow up. Like if you're growing it out on an agar plate, aerial would mean it would grow up kind of like a plant, or imagine it like a tree growing up from the dirt. That would be aerial. We use biochemical testing and molecular methods primarily to ID yeast. Yeast reproduce by budding or fission that involves maturation of a bud to an independent blastocondidium, or a daughter cell. The structure enlarges and the nucleus of the parent cell undergoes mitosis. A new nucleus passes to the daughter cell. The septum forms and the daughter cell begins to break free. So if you look over to the yeast, the side with the yeast, you see the parent cell is the larger cell, and this smaller bud, this is the daughter cell. So let's look at that a little bit closer on the next slide. So this is the process of a yeast cell budding or reproducing by budding. You start out with a single parent cell, and the parent cell starts to form the bud, and the new bud starts to pinch off, or the septum begins to form, and once it's pinched off, there's a chain of daughter cells that can also form from this one parent cell. So we're still comparing yeast with mold, but let's talk about mold. Mold is characterized by having hyphae. Hyphae are these long finger-like projections that you can see over here in the image of mold. Most have a fuzzy or woolly appearance because of the formation of micella, which are made up of long tube-like structure called hyphae. And like I said, we can see those over here in the image. Hyphae can be characterized as either septate or aseptate. Septate is where you will see cross walls perpendicular to the outer wall of hyphae, and we'll see this on the next slide. Aseptate or cenocytic are the absence of those separations, and again we're going to see that on the next slide. Also with molds, the mycelia or hyphae are either characterized as vegetative or aerial. Aerial, we've already mentioned, is from above the surface, giving it a fuzzy appearance. Remember, that's like the plant growing up from the soil. Imagine a tree coming up from the soil, so it's projected upward. Vegetative would be extended downward into the medium to absorb a nutrient. So think of like roots going down into the soil. So here is examples of hyphae of fungi, and we're talking about the hyphae of mold, and this is comparing septate versus aseptate. The septate is going to have separations and make the hyphae look like it's divided up into little compartments or different sections. Okay, so this is septate if it's divided up. If there's no separation, no cross walls, this is known as aseptate. But both of these structures, these finger-like structures, are both hyphae. It's just one is characterized as septate and the other is aseptate. So the microscopic appearance of hyphae can help aid us in identification of the particular organism. So some common words used to describe hyphae are either antler, which is an example right here. Then there is racket, which it looks like it has little bulbs, kind of like a tennis racket. Also there's rhizoid, which is here on the end. And then there's spiral hyphae. Our hyphae can also come in different colors or different shades. We have two categories. We have hyaline and dematiaceous. Hyaline is hyphae that is non-pigmented or lightly pigmented. We have the dematiaceous, which is hyphae that is darkly pigmented due to melanin in the cell wall. Depending on the amount of melanin present in the hyphae, it could appear anywhere from pale to dark brown to almost black. In mycology, we often use Fontana-Masson stain to determine pigmentation because it stains melanin. Dematiaceous hyphae appears brown, and hyaline hyphae stains pink to red. Now we need to discuss the difference between dimorphic and polymorphic. Dimorphic fungi include a mold phase and a yeast phase. The yeast tissue state is seen in vivo, or in vivo means within the organism, when the organism is grown at 37 degrees Celsius. Remember, 37 is around body temperature, with increased CO2, or also in vitro outside the organism. The mold phase is seen when the organism is grown at room temperature, that's about 22 to 25 degrees Celsius in ambient air conditions, so out in room. Polymorphic fungi have both yeast and mold forms in the same culture, so it can occur despite growth conditions. So the temperature doesn't matter as much for the different forms, whether it's yeast or mold. Now we need to talk about the difference between sexual and asexual reproduction for fungi. We're first going to discuss sexual, also known as perfect reproduction. Fungi that reproduce sexually are also known as telomorphs. They require the joining of compatible nuclei followed by meiosis. If you look at our image over to the right hand side, this top portion is asexual reproduction, and we're not really focusing on that. We're looking at sexual reproduction. You have two cells from different mycelia joining together to form a diploid zygote, and then meiosis is happening to form spores. So this is the process of sexual reproduction for fungi. These are all examples of sexual reproduction in fungi, and it just depends on the species and what type of cell they produce is what they're called for sexual reproduction. First we have the ascospore, which is a sexual spore. It's contained in a sac-like structure called an ascus, and the ascus is just a sexual spore where spores are found in sacs on the fungi. Then there's the blastidiospore, and this is a sexual or reproductive spore, and it's formed from blastidium, and you can see an example of that right here in the image. Then there's the zygospore, which is a cell arising from fusion of two gametes. The hyphae fuse to form protective zygosporangium. Now let's discuss asexual imperfect reproduction. Fungi that reproduce asexually are known as anamorphs. Asexual reproduction results in the formation of candidia. Remember, they're spores from the tip of the hyphae following mitosis. Asexual reproduction is carried out by fruiting structures known as condygenous cells, and there's many different types. In the clinical lab, most mold IDs are based on the structure formed as a result of asexual reproduction. So if you look over here at our image for asexual reproduction, you have these mother cells or parent cells that are able either to divide and make daughter cells, or the bud is able to form a daughter cell. That's a form of asexual reproduction. Or if the hyphae is able to be split off and produce a whole nother organism, that's a form of asexual reproduction. Here are some examples of asexual reproduction cells, and some of the cells will have examples over on the right hand side on the image. There's Phylida, which is a vase-like structure that produces phylocondidia. Next is Annelid, is a ring structure that produces anelloconidea. There's Arthroconidia that produces arthrospores. There's Blastoconidia that produces blastospores. There are sporangiospores produced inside sporangium. And finally, there's Chlamydioconida that produces chlamydiospores. Disease caused by fungus is known as mycosis. It can be superficial or cutaneous, subcutaneous or systemic. But let's first start off by talking about superficial. Superficial infections can include the hair, skin, nails, and cornea. These are infections confined to the outermost layers of the skin or hair. Cutaneous infections are defined as fungal diseases of keratinized tissues of humans and other animals, though dermatophyte species are capable of causing similar infections. These diseases are usually named with respect to the portion of the body that they affect. Subcutaneous infections usually involve deeper skin layers, maybe involve the muscle, connective tissue, and bone. Characteristic clinical features include progressive, non-healing ulcers and presence of draining sinus tracts. Systemic infections or disseminated mycosis infections are ones that affect internal organs or deep tissues of the body. The initial site of infection is frequently the lungs, and then it disseminates to other organs, including the skin. Chronic cough and chest pain might accompany systemic infections. This concludes part 1 of our mycology lecture. I will see you over in part 2.